Now, to be clear, when I say amplification, you're going to hear me talk about that a fair amount. It's not that we are doing any sort of PCR. Thank you, gentlemen. It's not like we're doing any sort of PCR where we're amplifying the actual RNA that's in the sample. Instead, we're just boosting the signal. Cool.
So next slide.
All right, awesome. So yeah, so RNAscope 101, here we go. We are up and running. I'm just going to run you through an introduction of the RNAscope technology, how it works, why it works. I'm going to give an overview of the assay that, for those of you in the workshop, we're going to be doing, which is the red chromogenic singleplex assay. I have a couple of slides that talk about our other assays. We have a couple of different flavors of RNAscope. Depending on what application specifically you have, what type of tissue, you might be interested in one or another of the assays that we have. And then, yeah, I did want to touch on IHC because that's a question I get a lot. And then if you have any questions, I mean, shout them out. This can be super informal. If you have questions as I go along, please feel free to raise your hand and ask. I may get to it later on in the presentation, but definitely I'd rather you shout out rather than forget your question. So this is my email address. For those of you who are listening in remotely, if you can't ask a question or virtually raise your hand, feel free to shoot me an email. It's emily.martersteck, sorry for the long last name, at bio-techne.com. And then I threw Jane's email up here as well, because if you have questions about ordering or pricing or which assay would be right for you, Jane is an excellent resource, and she really is fantastic. Okay, so diving right in, if you have an in situ hybridization background, you are familiar with this overall process where we start by digesting our tissue enough that we can get probes in there to penetrate and hybridize to your RNA target of interest. And then with RNAscope, we come along with these funny looking double Z paired probes. We then do this amplification series where we build sort of a tree-like scaffold on top of these probes. So the probes are not directly conjugated to a fluorophore or a chromogen. We're sort of constructing this tree on top of the probes, onto which then we can throw a ton of label. So that's where the signal amplification comes from, is we basically just build this thing and we can throw so much label on it that you're able to visualize individual RNA transcripts. So what RNAscope data at the end of the day looks like are these punctate dots. And wherein each dot corresponds with one RNA molecule, one RNA transcript. And then you can quantify these. So you get the single RNA molecule resolution. You also get the tissue morphological context versus a PCR where you kind of, bind and grind or grind and bind. You can look down at your tissue on these slides and see where in the tissue your target is expressed geographically.
Do you have to have the RNAs in the cytoplasm, or is this also data visualize it while it's being transcribed?
Good question. No, it's designed to pick up cytoplasmic RNA. Now, there's nothing to stop our probes from binding to the transcript, the target transcript, wherever it may be. So if it is a nuclear transcript, for instance, like a pre-microRNA, I don't know, some of these, if you have an AAV that you're using, you could in fact see RNAscope signal in the nucleus as well. Typically we don't like to because we worry about picking up DNA. So yeah, if you're looking at an mRNA transcript, most likely it's going to be cytoplasmic. So this is what these probes look like. You got two Z's where you have on the bottom a region that binds to your RNA transcript. Each Z, this bottom portion here, is roughly 25 base pairs long. You need two Z's to come down and bind exactly adjacent to each other on your transcript, such that then a total of 50 base pairs is being bound by your Z's. But they're binding all along your transcript. So when we design a probe, we take usually about a 1 kb length of sequence. So each RNAscope probe is actually a pool of these Z pairs that are going to come down and carpet your transcript. So we like to have about a 1 kb region to design a probe against. We can do shorter sequences. You just have fewer Z's available to bind your transcript, which isn't necessarily a problem. The top portion of the Z then is this pre-amplifier binding site. This is when two Z's bind together, it forms this 28 base pair sort of platform. And we're going to come along with a pre-amplifier molecule next. This is a prettier picture of this. So again, you've got your, if the gray part is your RNA transcript, each Z comes down and binds to a portion of that transcript. Then the top portion is that pre-amplifier binding site. After that, oh, and I should mention, so it's possible that you would get, this 25 mer somewhere else in the transcriptome. It's a short sequence, that's possible. But if you only get one Z of the pair that binds onto your transcript, this pre-amplifier hybridization event is not going to be stable enough with just the one Z. So that pre-amp molecule is going to get washed off in subsequent wash steps that we do. So that's where your specificity comes from. You shouldn't get binding off site from your target of interest.
So
Mhm.
you need 1,000 bases.
Yes, the question is, if you need 1,000 bases, what do you do with smaller transcripts? RNAscope probes will work on sequence lengths of down to 300 base pairs. Below 300 base pairs, so if you have like a pre-microRNA, for instance, you're going to want to move to our BaseScope assay. And I have a couple slides about that later on. As long as you get 6 pairs, 6 double Z pairs that bind somewhere along your transcript, you're going to see a dot. So if you have 20 available to you, you stack the deck in your favor of getting at least 6, right? After 6, it's just gravy. You get a slightly larger dot. You need 6 to see a dot in the first place. Does that make sense? So after the pre-amp molecule comes along and binds our probes, we come along with amplifier molecules that are sort of the branches of our tree, onto which we then come along and put labeled probe onto. For the workshop folks, we're going to be doing a red chromogen for our visualization of our probe. And then, yeah, as I mentioned, once 6 double Z pairs come down and binds anywhere along your transcript of interest, you see a dot. So this is our brown chromogenic assay. I'm not actually sure what type of tissue this is that we're looking at, but you can see all these brown punctate dots spread out all over the tissue. That's what your RNAscope signal looks like. This is the multiplex fluorescent assay. So we're actually looking at 2 different genes here at the same time. And I just wanted to touch on, you can get a highly expressed transcript that you might be visualizing, in which case the dots all sort of overlap and start to cluster together. So that red blob there, that's still a bunch of individual punctate dots, but there's just so much of them in that cell that they sort of overlap and start looking like a blob. And then just as an illustration of this, the power of this sort of double Z design. So at the top upper left corner there, you have both Z's, sort of a left-hand Z and a right-hand Z, our typical RNAscope probe. This is looking at 18S RNA. And then the upper right panel there, no target probe at all was applied to the slide, but then the rest of the RNAscope assay was performed. Bottom left is just the left-hand set of Z's, and the bottom right was just the right-hand set of Z's. So as you can see, you don't get any signal if you just have one or the other Z. So this really is, it's a cool assay. The design of these probes allow us to be really specific with what we're trying to visualize and pick up. Just to go over what I just told you, sensitivity comes from the ability that we have to amplify our signal such that we get down to single transcript resolution. It's specific because we need 2 Z's to come down in tandem, bind exactly adjacent to each other before we get that signal amplification. You get to look at where the gene is expressed in your tissue, and you can quantify this. And antibodies, they really are great, right? Because it's a much easier incubation when you're doing IHC. However, we can design a probe against any gene in pretty much any tissue. If you have an annotated transcriptome for, we have mollusk probes, we have cow probes, we have the traditional mouse, rat, human probes. But kind of whatever you are interested in studying, we could probably make a probe against it. Question was, yeah, am I going to talk about long non-coding RNAs? I wasn't planning on it. Sure. So that's a good point. Like there are some applications we just don't have good antibodies for at all. GPCRs is a big one, for instance, long non-coding RNAs. And this would be a really great way of looking at that. Secreted proteins is another one. You can stain for secreted proteins, but it's just sort of, you're just going to get a wash of signal everywhere. Versus if you have an RNAscope probe against that transcript, you can see where exactly, like what cells are producing that protein, even though it travels far away. So here are all of the flavors of RNAscope and BaseScope, as I mentioned, that we have. And today we're doing this RNAscope probe red chromogenic assay, that first column there. We also have a duplex chromogenic assay where you can look at 2 targets at the same time on the same slide. And you can do red and green chromogens for that. Then our fluorescent assays, there's 2 different flavors of those. The third column from the left, the RNAscope Fluorescent Multiplex assay, that was the first iteration of the multiplex fluorescent assay that was released. This is appropriate for fresh frozen tissue, but then we have folks that want to do fixed tissue that's been fixed and then frozen, or FFPE samples, formalin fixed paraffin embedded tissues. So they came along with the multiplex V2 assay. There's an additional amplification step in this assay because when you fix a tissue, you run into major autofluorescent problems, especially in the green fluorescent channel. So the V2 assay takes advantage of the enteriomide signal amplification system. So you get the TSA fluorophore dyes from PerkinElmer to use with that assay. And then far over on the other side, the BaseScope assay that I mentioned, where BaseScope is appropriate for short sequences, so less than 300 base pairs. You can actually look at exon junctions. You can look at, you can validate a knockout. There's a bunch of different cool things you can do with the BaseScope assay you can't do with RNAscope. And if people have questions about that, I'm not going to go too much into detail about that right now. Feel free to email me though, and I do have a couple slides I think at the end. So, okay, what does the assay actually look like? Kind of an overview, this is the FFPE workflow where we have sort of a couple of different portions of the assay. The pre-treat part, then we come along with our probe, hybridize it, and then we do that amplification series. And lastly, we throw a chromogen, or if you're doing the fluorescent assay, you're gonna throw on fluorophores. So up top there, the first step with FFPE tissues, you have to melt the wax out of there. So for you workshop folks, we started baking the slides that have FFPE tissue on them already. So they're in our 60-degree oven now. We're gonna come along with a peroxide block that helps with background. After that, we do 2 things. Again, this is FFPE tissue. One is an epitope retrieval step, and the other is a protease digestion. So we have to allow our probes to get into our cells so that they can bind and hybridize our targets. So we do a heat-induced epitope retrieval where we're boiling tissue in a target retrieval buffer. And then we come along with a protease to digest some of that extracellular matrix and punch holes in cell membranes. Because although RNAscope probes, the part that actually binds your target is a short sequence, the probes themselves are physically fairly large, as are the amplification molecules. That pre-treat portion of the assay is the only part that you actually need to optimize depending on what your sample is. Different sample types want different strengths of digestion and pretreatment. If you have, embryonic mouse tissue, it's a delicate tissue, you're not going to need to do much epitope retrieval or a long protease digestion. Versus if you have, an adult mouse liver, we know that to be a very tough tissue. So we might have to go longer with digestion to allow our probes to get in there. And that's the only portion of the assay you actually need to optimize though, because once you hit the probe hybridization step, there's an A there on that workflow. After that, the assay is identical. So it's cool because any RNAscope probes you get from us will behave the same way. As soon as you know for your particular sample what pre-treatment conditions you need to make it work, you should be good to go from then on, as long as your samples are prepared very similarly to each other in terms of length of fixation. For fresh frozen folks, I know I have a couple of you today, no need to depair. There's no epitope retrieval required. That epitope retrieval step is to reverse the fixation crosslinks you get when you use formaldehyde fixatives, which we like because it helps lock the RNA into tissue, so you know that there will still be RNA left to visualize. But you have to heat retrieve to break up those crosslinks to get our probes in there. So fresh frozen folks, there's no fixation step, you don't need to depair, and you don't need to do heat-induced epitope retrieval You just do the protease. So people ask me a lot, what sample prep should I do? Should I do FFPE? Should I fix and then freeze? Should I snap freeze? It's totally up to you. RNAscope will work as long as there's RNA left in your sample. Fresh frozen, the workflow is shorter because you don't have to bake or do your heat-induced epitope retrieval step. But it's totally up to you. Depends on your experiment.
So for all these pre-treat steps,
Mm-hmm.
do you recommend using RNA-free water for preparing fixatives or alcohol?
Yeah, good question. The question was, do you need to use RNase-free reagents in the preparation of these materials? No, which is kind of crazy. If you have an in situ background and you were super careful about making DPC water and using RNA sap and keeping everything clean and making sure no one sneezes on your samples, it doesn't hurt to use RNase-free water or RNase-free preparation, but we don't need it because RNAscope probes are sampling your RNA target in these little bite-sized chunks in 50 base pair pieces. So even if you have some RNA degradation or fragmentation, we should still be able to pick up the signal. So it's crazy, but yeah, in the workshop today, we're not going to be doing RNase-free conditions. Again, it doesn't hurt. And if I have a sample type I come across where someone's not getting signal, that's one thing I might recommend is, hey, especially in your sample prep portion, like when you're collecting your tissue, use RNase-free water. Be a little bit more careful. Wear gloves, but it's not necessary.
Are there issues with RNA degradation from FFPE?
Sure.
Normally, sequencing of hair,
Right.
Fragment anyway.
Mhm.
But are your signals sort of used, or is the RNA basically still in the same location because it's fixed? So,
Yeah, the question was, do you worry about FFPE affecting your ability to visualize the RNA and where it is in the tissue, fragmentation? Not really. And that's because, again, like fixation and FFPE preparation does fragment your RNA. But again, we just need 50 base pair chunks to be intact. And so as long as you have 6 pairs of RNAscope probes that are able to find chunks that they can hybridize to, you're going to get dots. And the difference, a big difference between FFPE tissue and fresh frozen tissue, for instance, is you actually do kind of sacrifice some tissue morphology with fresh frozen tissue because you've done nothing to really, hold your tissue together, and we digest the tissue. So sometimes if you do a side-by-side comparison, same target probe with fresh frozen versus fixed frozen or FFPE tissue, the fresh frozen tissue looks a little bit more digested, like the signal looks maybe a little bit more diffuse. But there are things we can do. We can back off on protease digestion to try and mitigate that.
I guess my question really is,
Sorry.
you get an artificially high signal for count, right? So if you're looking at a specific target,
Oh, sure.
your RNA takes big RNA and probe it.
Mm-hmm.
You see 3 smaller dots.
No.
Accidentally count instead of 1.
Yeah. The question is, if your RNA is fragmented, do you get artificially high signal? No, because we need at least 6 to bind before we even see a dot at all. And so if you don't have at least 6 pairs, you're not going to see a dot. And again, like the probe is designed against a specific sequence. So we only have 1,000 base pairs to work with. So in that space, you shouldn't, like the RNA shouldn't be too much fragmented and moving around the tissue. In sequencing, you worry about ambient RNA being released into the solution. That's not so much of a big deal here. Does that answer your question?
Yeah.
Okay. This is what the stuff looks like. When you buy the RNAscope kit from us, you're going to get a bunch of pretty colored boxes. Your pre-treat reagents, that's that hydrogen peroxide I mentioned as a blocking step, your protease, and your target retrieval buffer. The target retrieval buffer comes at a 10x concentrate. So first thing we did this morning was dilute it down to 1x. The detection kit has your amplifier reagents that you need to be able to visualize your signal. The chromogen as well. If you're doing the fluorescent assay, the V2 assay, you would need to, again, get the TSA or opal dyes from Perkin-Elmer. And then wash buffer. We have a fair amount of wash steps in there that make sure that you don't have background issues. And that comes at a 50x concentrate. A quick tip, if you ever see precipitate in your wash buffer, just warm it up to 40 degrees. The precipitate should go right back into solution. That heat-induced epitope retrieval step I mentioned, if you have fixed frozen tissue or FFPE tissue, there's two ways to go about doing it. One, you simply boil the target retrieval buffer in a beaker, lower your slides in there. The other is with a steamer, which is what we're going to be using today. The steamer is just a little bit more gentle on the tissue, so I prefer it, but if you don't want to order a steamer, I think it's like $30 on Amazon. You can use a hot plate in the lab and a beaker, doesn't matter. And then the high BZ oven from ACD. This is actually the older version of the oven. You'll see both in the lab today. We also have the newer version. I mean, any oven should work, theoretically. Our probes and our AMP molecules hybridize at 40 degrees. If you have an oven that is able to keep 40 degrees, you know it's 40 degrees, and it's super stable, and it also keeps everything humidified, your RNAscope assay will work. I like this oven a lot, though. I've had a case where someone's assay wasn't working, and the second we switched them to this oven, it worked beautifully. And I know we have a loaner program. Jane, we have a loaner program that.
So if you are doing a demo and you don't have an oven, we can always send you one. You up to date, and then at the end of it, if you want to compare another you have, this one, so do that, and back at the end. So we started sending a loaner because that was very helpful.
So for those of you listening remotely, Jane just mentioned that we have this loaner program. We'll send you an oven free of charge for 60 days. You can keep it. You can compare to whatever incubator you have in the lab to make sure that that one will work. Or you could keep it and convert it to that being your oven that you run from then on. What's the price? I actually don't know. What's the price? Thanks, Jane. The question was the price of it. And so the refurbished version is less expensive than if you buy it new from us. Tips for success when you go to run your assay. First of all, we have these protocols that we know work. So a lot of time, I mean, we're scientists. We like to be creative. We like to mess with things. Like knock yourself out. I'm not going to tell you not to, but the assay was designed in a specific way. And so start with the assay as the protocol is written. And then if you need to make modifications, run your controls, make sure that anything that you change is still going to work for your assay. Really, though, the only piece that you need to mess with is that pre-treatment portion of the assay. And then the control probes are really helpful. The first thing you should do when you first get your RNAscope stuff in is take your samples and run a positive control and a negative control. The positive control is a housekeeping gene. It will tell you whether or not you have good quality RNA left, whether you have enough in there that you should be able to visualize your target with. And then the negative control probe, you should not see any staining on your slide that has the negative control applied to it. And I have pictures of that in a second. And then there is a fair amount of stuff that you need from us. So here's pictures of the control probes. Our negative control probe is DAP-B. It's a soil bacterial gene. So this should not be present in any mammalian system. If you go outside and drop your slides in the dirt, you might pick some up, but don't do that. And instead of using like a scrambled oligo or just a blank probe, we are probing for a real gene, but you shouldn't see any signal. As compared with, so here's one of our positive control probes, PPIB, it's an isomerase. It's a medium to high expressor in almost all tissues in most systems. You should see a ton of signal here. We have a couple of different flavors of positive control probes. We have one that's an even higher expressor than that. We have a lower expressor, polar 2A. If you have a target that you know is a pretty low expressor, maybe go for the more lowly expressed housekeeping gene as a better positive control. But this just makes sure that if you have RNA there, you'll be able to pick it up. And then, so here's just one example of a target probe. And this is the red assay that we're running today. So IL-6, you can see it's pretty specific to certain areas of the tissue, but the tissue looks really good from the positive control perspective, and the signal looks good in the target. And so we have this semi-quantitative scoring guideline. it just kind of as guidelines for what you should see. So the DAP-B negative control should be a score of 0. You really shouldn't see any dots. You probably want a score of at least 2 for most tissues that you would run, so that you know you have RNA left that your target will pick up. But then once you get up to 3 and 4, especially a score of 4, you start to see those clusters happening, and that just means you have a ton of signal. Tips for success. Here's some important guidelines that you should keep in mind. There's a bunch of different AMP steps. Make sure that you do them all and do them in the right order. Otherwise, the assay won't work. In between each different AMP application and wash, flick the excess reagent off, but you do not want to let your slides dry out in between steps. If you do, you probably won't be able to get signal. It's because these are modified DNA oligo molecules. So if your slides dry out, they won't be able to hybridize. And then for this manual assay, we're going to draw a hydrophobic barrier around your sample on your slide. Make sure you get a good barrier that it'll keep the liquid on the slide, nothing leaks out. The pen that we recommend, it's the image pen. It's awesome. It should hold up throughout the entire assay, pretty much anything you do. So yeah, definitely I recommend that one highly. Yeah, don't alter the protocol. It works, I promise. And then, yeah, you want to make sure that you have an oven that maintains humidity. Fresh reagents is also important, especially for PFA. When you do your fix with 4% PFA, make sure you do make it up fresh that morning. PFA tends to drift. And so if it's old PFA, you're not going to get good fixation. For the red assay specifically that we're doing today, a couple other considerations. We recommend EcoMount. Vectomount will also work, but this red chromogen is sensitive to ethanol. So if you have a mounting media with residual ethanol, it'll start to make your chromogen look really bad and spidery. And instead of at the very end of the assay, we're not going to do an ethanol dehydration. We're simply going to bake the slides. It says at least 15 minutes. I bake them for 30 minutes to make sure they're dry before cover slipping. For the brown assay, we've noticed that in some tissues that have a really high endogenous alkaline phosphatase activity, like kidney or adrenal gland, you kind of get a little bit of a red haze, a pink haze background with the red assay. So here's the chromogenic brown assay. That's going to avoid that problem. But on the flip side, lung tissue or skin samples that have brown pigment to them, melanin, or in lung tissue, you sometimes see these dark brown carbon deposits. Maybe you'd want to use the red assay instead in order not to confuse your RNA signal with the pigmented stuff that's already in your tissue. And we say that the brown assay is not recommended for combined ISH and IHC. That's because if you're doing ISH followed by an IHC, you want to do your RNA scope first, then your IHC. And we kind of worry this brown assay uses the DAB chromogen, and DAB is physically fairly large. But honestly, I have people that follow this assay with IHC and it's fine, but just a consideration. And the duplex assay, I'm not going to go into this too much today. Multiplex fluorescent, you're able to visualize up to 4 targets in one sample at the same time. This is what some of that data looks like. I'm going to speed up a little bit just because of time. Okay, so I mentioned the pretreatment portion of the assay. This is where you might need to mess with it a little bit in terms of timing, depending on what your sample is. So this is what sort of an optimal digestion looks like, the balance between digesting and over-digesting and under-digesting. You need to get enough tissue digestion that our probes can get in there, but you don't want to go so far you get to this point of diminishing returns where your tissue starts to look like mush. So this is good. On the right there is the negative control, nice and clean. I think the negative control is easier to look at when we're talking about morphology of tissue. You can see the nuclei aren't, they're a little bit cloudy, almost like fuzzy. But then on the left here, PPIB is again, one of our housekeeping gene positive controls. You get really good signal as compared with, this is an over-digested sample. So you've gone too far. You've either boiled it too long or you used too harsh of a protease treatment. The tissue, again, especially looking at the DAP-B control on the right there, looks like mush. And your signal starts to get a little bit more just diffuse. as compared with, let me flip back real fast. So this was optimal. You can see that nuclei are just a little bit more intact. The hematoxin's a little darker. And then this just looks like mush. Versus under digestion, here we haven't gone quite far enough. So you can see the tissue on the right there. It's still really intact. The nuclei are very dark and well defined. But the PPIB, you barely get any signal. You can see a little bit here, some dots. But we haven't gone far enough with this tissue. So again, just back at the optimal. So this would be under digested tissue. And that also, we have these fixation concerns. We recommend to fix somewhere in between 16 and 32 hours if you're doing fixed tissue. Longer than that, you can have problems with the assay. You're not going to get as much signal. Now, accordingly, if you have an overfixed sample, you could just digest a little bit harsher and hopefully rescue some of your signal. But it's a good idea to follow the guidelines. 16 to 32 hours is awesome. So here on the left, this is PPIB, on the left. And so 24 hours fixation here as compared with 3 weeks on the right. You can see way more brown signal here on the left than the right. And because that was fixed for, 3 weeks is a really long time for a fix. Now, I do have folks that have overfixed tissue because they have a collaborator and they get these samples from the clinic and they have to work within the confines of their collaborator's protocol. There are things we can do to try and help boost the signal in terms of the pre-treatment optimization, but if you have the choice, definitely stick within that 16 to 32 hour window. Fluorescence, one thing to mention is that we do have autofluorescent issue in fixed tissue, especially in that green channel. So if you ever run the multiplex fluorescent assay, something to keep in mind, put your highest expressing target in green. You're gonna boost your signal to noise ratio that way. Again, I'm not gonna go too much into this. Also, multispectral imaging software, if you have access to some of those capabilities, that's also incredibly helpful. Does anyone have specific questions about the multiplex fluorescent assay at this time? If not, I'm just gonna kind of scooch on by some of this stuff to IHC. So as I mentioned, you can follow your RNA scope with IHC. Please do your RNA scope first because we do need the RNA quality to still be intact. And if you do your IHC first and then try and come along with doing an in situ, you might not have RNA left in your sample. It probably has degraded at that point. But one thing to consider is that the RNA scope pre-treat portion of the protocol, where we're heat retrieving with a boiling and buffer and doing a protease digestion, can sometimes ruin your epitope of the antibody that you want to use is trying to recognize. So first, just sanity check, take a sample, run it through the RNA scope pre-treatment portion that you've determined works best for your sample, and then skip RNA scope, the actual probe and amps, and go straight to your IHC protocol to make sure your antibody staining still looks okay. So for instance, with cell membrane bound proteins, those especially are affected by the RNA scope pre-treatment. So you may have to then back off on pre-treatment, sacrificing some of your RNA scope signal in order to rescue your antibody staining. So that's another place where you might have to just empirically determine what's going to work for you. It can work though. Here's an example. This is a non-small cell lung carcinoma FFPE tissue sample. And this, I apologize, it looks way better on my laptop than it does on the projector screen. But we have 2 RNA scope targets in green and red there. And then the CD8 in white is antibody. Oh, thanks, Doug. So CD8, if you're familiar with that antibody stain, there's usually quite a bit more of it. It looks more like a halo. After RNA scope pre-treatment, you can still see it. But the IHC, the antibody staining, is somewhat diminished. So just do yourself a favor, just sanity check that your IHC will work in RNA scope pre-treatment treated tissue before combining the two. And then our website, go to acdbio.com. There's a ton of materials on there. There are recorded webinars, there's training videos, frequently asked questions. All of the user manuals, any documentation, the protocols that you're going to need are on the website. It can be slightly hard to navigate, in my personal opinion. So also feel free to email our support because we have a team of tech support scientists that is their job, is to help you with RNAscope and making it successful. And also, yeah, I'm your local FAS. So if it gets to a point where you need on-site technical support, you would see me again. And that's all I've got for you today. I will take any questions you might have.
How do you go about selecting the genes? You have to list the genes, you create a sequence, how does that?
Exactly. The question is, how do you go about designing your probe, basically, right? So we have a probe design team. They're all PhD scientists, and this is what they do: is whatever sequence you're trying to visualize, send them either the specific sequence or the gene accession number, and they're gonna use their fancy schmancy top secret algorithms to design the probe, because what they're looking for is portions of that gene that don't have high homology to anywhere else in the transcriptome. They take GC content into consideration. They don't want areas of high repeats, all that kind of stuff. And then they're gonna pick an area along that gene that you're trying to visualize where they can design the probe. Now, as I mentioned, it's a pool of these double Z pairs. So on average, we have 20 pairs, but depending on your actual sequence, there might be fewer, there might be more. There's a probe that has 29 pairs. But if it's a gene that's a little bit trickier in terms of areas of high homology or GZ content, whatever, you might have fewer double Z probes that they could design. But again, as long as you have 6, so a 300 base pair sequence that we can work with, you're going to get signal. Yeah.
For small, like less than 100.
Yeah, the question is, what about small RNAs? So that's a BaseScope assay. I don't have a good, really awesome picture of it. So I told you RNAscope probes, we need 6 pairs to bind somewhere along the transcript before we see 1 dot. BaseScope is different. BaseScope probes are either 1 double Z pair or 1 to 3 double Z pairs. So those of you paying attention, I told you we need 6 to bind before you see a dot. BaseScope, you have fewer double Zs, but then we just build a humongous tree on top of them. So you have additional amplification steps such that you can visualize even just 1 double Z probe. So if you have a pre-microRNA or a really short sequence, the question is mature microRNA. I don't believe that we have capability to visualize those yet. Yeah, we need at least 50 base pairs. Yes, but here's just one application for BaseScope. If you have, if you come along with CRISPR and you're splicing out a portion, you can design a probe against like the wild type probe, or we can design, a probe against the new junction that will be formed when you splice that portion out. Alternative splicing would be another application of BaseScope. So short sequences, we can even use BaseScope to interrogate point mutations. However, that is a services project. So we have an in-house team. If you're interested in looking at point mutations, contact Jane, because she can potentially get you set up with our in-house services group that will run those experiments for you. That's just a trickier application, so they want more control over how that experiment is run. Does that answer your question? That's true. Jane mentioned we have a couple of validated probes. With point mutations, they just have to work harder to design a probe that's going to work. And so they design a couple of different probes, and then they, I think, make cell lines as controls to validate the probes. That's a ton of work. Our RNAscope probes and BaseScope probes generally are validated in silico, just in the way that they're designed. So it's a tricky application, but yes, we do have some then validated single point mutation probes available on the catalog, which is super cool in my opinion.
How is it expressed? Because if you're looking at an FFPE, where you've got a 4 micron or whatever slice, it's not a cell. So what do you?
Yeah, good question.
Relative signal, like what's the way to quantify?
Yeah, the question is actually quantifying RNAscope data. What is it? It's the number of dots. So if you are sectioning tissue, and you have thicker sections. So for FFPE tissue, we recommend, I think, 5 to 7 micron sections. Fixed frozen, we recommend somewhere more in the order of 10 to 20 micron sections. The thicker section you have, the more dots you are going to see because you're capturing more of the cell and thus more transcript that's present there. But in terms of, what else was I going to say? Ask me the question one more time. I had another point to make.
So it was really about how you sort of report the
Yes,
quantity.
thank you. Yeah, you will see some dot size variability. There's no biological relevance there except that you get a larger dot if you get more double Z pairs that bind on your transcript. You need 6 to see a dot at all. If you have, between 6 and 20, you get a larger dot. But yeah, in terms of quantifying, you're counting dots.
And so on top of that, so I mean, I can see that how the software can say, okay, here's the average dot per nucleus, and how are you, that's really what I'm getting into. And then how do you determine in cell types, right? So if you've got a non-small cell lung cancer, you're gonna have a lot of different cell types in that tumor. So how are you assigning dots?
Yeah, we actually have a couple of webinars that go into this great detail. We typically do an H-score for different sample types, and then across your study. So I think I can send the link to some of those as well. But you're right. One of the nice things about getting morphology is you may see very different areas of expression, like in this tumor, and you'll be able to sort of quantify with a relative expression.
And we have also, in addition to those webinars, Halo is a software that a lot of people use. We have an ImageJ tech note, like if you have Fiji, you can use that as well. Other questions? And again, feel free to email me if you have additional questions, those of you listening remotely. But if that's all for now, I think I might take the folks that are doing the workshop. Yes.
What do you do for signals that saturate? You showed some of those that are just blobs.
Yeah, exactly. Question is, what do you do with signals that saturate? So yeah, at a certain point, you can't quantify individual dots, right? Because the dots are so overlapping, you do get that blob, that clustering, which is why if you remember, our semi-quantitative scoring kind of caps out at 4, because above that, it is that cluster. I mean, I think, yeah, Jane?
We had a couple of instances where there was something so highly expressed that instead of putting 20 VG people together, you may only have a 1 VG in that for that particular target for the mallet. But if you look on the mallet, it's all like 1 VG or 2 VG stuff too. So just to be able to see a punctate dot rather than just a smear. But it's tricky.
Jane said we can design, if your target is incredibly highly expressed such that it's so overlapping you get a cluster, we could design, we can almost cripple the assay and design a very quote unquote short probe, so a 1 ZZ probe or a couple of ZZ probes. But yeah, I mean, with UBC, for instance, ubiquitin C, it's a very highly expressed housekeeping positive control probe we have. Yeah, it's just sort of a wash of color. And at that point, I mean, you'd kind of be looking at more of like a yes, no than a copy number. All right. That's all we've got. Yeah, again, if you think of anything else, shoot me an email, but we'll probably move into the lab because our slides are done baking at this point. Cool. Thanks, everyone.